Okay. Good morning, everyone, and welcome to our section on imaging genetics. And actually, we have 4 distinguished speakers. And our first speaker is Dr. Shen from the UPenn. He's a well-known figure.
So, thank you. Yeah, I can use this. Yeah. Thanks, Hongtu, for the introduction, and also thanks to the organizers for having me here. So I think it's been great to be here, and both the venue and the programs are really remarkable. I'm going to talk a little bit about brain imaging genomics and Alzheimer's disease. So we know actually the major challenge in Alzheimer's disease is like the. Do I need to start? Oh, already started. Okay. So I think there's no drug that can modify the disease, although there's some like recent development, but this is still a major topic, right? So that actually we have to find the genes and the proteins, right, as targeted and look for drugs. That's why we need to understand the genetic molecular mechanism of AD, right? But we know actually the major hallmark of Alzheimer's disease, right? So we have this ATN like system, and all these can be captured actually by imaging, right? So that's why we use imaging as phenotype to study genetics and the genomics, right? So Paul Thompson, I wrote this kind of a review paper in like five years ago on brain imaging genomics. We kind of try to figure out the methods. I reviewed the methods that can kind of find the relationship between these imaging genomics and the phenotypic outcome. And so we actually have all these biobank data available, right? The ADNI, UK biobank, and so these are like the 3 problems we discussed a little bit, and I just want to kind of solve. Yeah, here I'm going to like cover like one study for each topic. The first one is more like the heritability analysis. So since there are a lot of imaging measurement, right? But the thing is, we want to pick those kind of more heritable imaging phenotype, right? So that's why we want to study heritability analysis. We have like a lot of atlases, right? The Yale atlas, AAL, right? Most of those are designed actually based on the anatomical knowledge or functional study, right? But we want to build an atlas that's actually kind of optimized for genetics stuff, right? So actually, a lot of what I talk about is today was developed by Bingxin Zhao. So he's a PhD student in genomics and computational biology program. So here actually he developed this giant atlas. Basically, we're using a UK biobank. So the idea here is actually it's a big data, right? And then we have all these like voxel-based measurement, like from crystals that was equals like a ribbon snap, gray matter, white matter, a lot of voxels, and we did a kind of heritability analysis at the voxel level using LDAK, and then you can actually segment this heritability map, right? We use this 3D Gaussian model. So the result is, if you look at the segmentation result, so our region we defined actually at the voxel level, they are more homogeneous, and you can actually also calculate the regional level heritability. By the way, this is actually tested on an independent cohort, not the data used for building the atlas, right? So we compared the giant segmentation and watershed segmentation, and also existing atlas, the Muse atlas, also crystals developed that. So I think the regional measure is more heritable. And then we also did some GWAS polygenic risk score analysis. And using giant hybrid atlas, we can find like more hits. So these are some kind of, these are the voxel level heritability measure. So one is the heritability, the other the kind of the p-value, right? And then, so here are the regions we identified. And we actually did some kind of GWAS analysis, try to figure out the kind of the genetic landscape of these imaging genetic findings. You can see all these are chromosomes, and color indicate different regions. And so here are these the giant phenotype relationship with other phenotype, like the the height, weight, BMI, that kind of thing, right? And then you can also do some functional annotation for these imaging genetic findings and calculate the genetic correlation and the phenotypic correlation. So you can see they are kind of similar, but there's also some difference. Right? So hopefully in the future, we can use that to do imaging genetic analysis. So the next one I want to talk about is basically find these imaging genomic association using CCA, which is kind of a very popular method. I think Yu is the expert. He's going to talk about that also later in this session, right? So, but the most kind of straightforward way is you can just do a massive univariate analysis, right? This was like a work we did 15 years ago. So look at all these whole brain kind of imaging phenotypes, and also these are the whole genome, like the SNPs. So you can map the genetic effect onto the brain, and also look at the genetic association across the genome for certain phenotype. So we know like imaging traits are statistically more powerful because it's continuous, right? So we actually look at also other phenotypes, right? The quantitative traits, CSF biomarkers, amyloid measurement. So a lot of genes are identified that actually are missed by the traditional case control gene study, right? But so there are a lot of like impact, right? So we have these Enigma, Paul Thompson, right? So also the UK Biobank and Hongtu and Bingxin did a lot of analysis. They also shared the summary data. And these are very valuable resources. And we actually use that in a lot of subsequent analysis. From our ADNI study, we actually identify some interesting AD targets. One of those are used to build a mouse model, right? So these are something actually we can probably help advance the AD research. Another thing in terms of the statistics, like you can use the CCA, right? Basically, you can extract a genetic component, right? It's like the polygenic risk score, but kind of using a different strategy, right? And also an imaging component, right? It's just a weighted sum of these original imaging measure. Then we want to maximize their correlation, right? And then we want to reduce the overfitting, right? So add additional constraints like the lasso or sparsity. So here in this work, we add a preference matrix. Basically, if I know this imaging phenotype has this genetic basis, maybe I can mark in the preference matrix. So this is a model. And so here's some proof of concept analysis. We looked at some AD phenotypes and some like phenotypes which are not related to AD, like gene expression. So these are the cognitive measure and imaging fluid biomarker free surface measure. We know they are related to AD, right? And for the genetics, we have 12 like AD loci and 19 AD not relevant to AD, right? So these are some participant characteristics, and here are the results. We compared with the CCA, sparse CCA. These are the preference matrix guided, right? CCA, sparse CCA. So we trained the model. You can see these are the correlation using the training data, but we apply those correlations to the testing data, right? Now you can see CCA, CCR overfitting, right? So here we actually, ours actually works the best. If you look at the biomarkers, we have imaging, genetics. This is a CCA, SCCA. These are our models. Now you can see actually, so these are actually all mixed together, but our model, we can identify irrelevant features, right? Those are things with 0, right, in the center. So that's one thing. And another thing is, so for the CCA, you can link imaging with genetics, but you can also map these different modalities into a common latent space, right? So sometimes it's important you want to do this fairly, right? If you have a model only works for males, then you design say a drug, then it's very expensive, right? That's not fair for females. So that's why we want to generate some fair representation. So here, the idea here is like, so if you map these two modalities into here, so randomly, so you don't see, are not aligned, right? But if you use traditional CCA, the male group may align better than female, right? You can do stratified analysis. If you just align the females, the males are not well aligned, vice versa. What we want to do is we want to build this fair model so that the disparity are kind of minimized between the male group and the female group. So we have some algorithm and a model defined. If you look at our experimental results, so the evaluation criterion, we have two things there, right? One is actually the correlation, that's a CCA objective function, and others are the disparity error, the fairness measure. We take the maxima and also the average, right? So you can see, and the data sets we did is, this is enhanced, and this is another, like the UCLA data set, and the ANI, right? So you can see on the CCA part, the correlation, we lose a little bit, just very, very, very kind of trivial, right? But for the fairness, actually the performance gain is humongous, right? These are on different data sets. So you can see actually this model can really help, right? And the last one I want to talk about is kind of like I try to link the genetics, genomics, imaging, and outcome together. So what we did is like, yes, so this is the parts we try to figure out, right? This is more like multi-scale analysis. And the first thing we did is just use UK Biobank. We did an imaging genetics study. We have a lot of imaging genetic findings, right? And then we want to prioritize those, right? Because we're interested in AD-related outcome, right? So we use actually ADNI-GWAS analysis. We do a co-localization analysis, and you can figure out these triplets, right? Genetics, imaging, and outcome. And then there's still a lot, right? Then we did actually mediation analysis, right? So that we can further narrow down the scope. We actually identify entorhinal cortex is the imaging mediator that links the SNP with AD, right? And then the thing here is that we still have this omics modality missing, right? So what we can do here is we have a lot of valuable summary data, say from the GPEX, right? So the EQTL, then we can do some co-localization analysis, or actually also link the genetics with omics and the imaging, right? So and we actually identify a very interesting gene. So this BTBD16, and we did some functional annotation, try to see what's the relationship in terms of the anatomy, neural anatomy. We see actually it's highly expressed in cortex, right? And in the cell types, it's highly expressed in this oligodendrocytes cell types. So all these things are kind of related to dementia and amyloid. So that's actually reassuring. So so if you put everything together, we can actually, the finding is we identified a SNP that can regulate the expression level of this gene. This gene is highly expressed in the cell type and also in these brain region. The region is happen to be an AD biomarker. So we can link all these together. So if you see the evidence at different level, that's actually reassuring, right? Yeah, I think so. Basically, that's it. And then we need to talk a little bit about the future direction here. So I don't know how many minutes I have. Okay, all right. So we have like 3 questions, right? So what are important research problems, and also how to use statistics to improve the data processing or in sharing, right? And then how the methods can make an impact, right? So just 3 topics here. What I'm thinking about is just some example. Here is the heritability heritability analysis, the atlas. This is more like a foundation model, right? You use a big data set, create something that can be used for other studies. If the data is big enough, maybe it's more generalized, can be used for different kind of applications. And so we're actually using statistical methods to generate these atlas, right? But you can also use AI foundation model. And if you really have something which is very different from all the distributions in this the big data set, you can actually fine tune using your own data, right? And in terms of the applications, we can actually use these to identify new AD biomarkers, new AD subtypes. The goal is you want to learn a representation, right? That can help you better accomplish the subsequent tasks. And for the CCA, it's multiple modality, right? It's multi-modal AI. So you can find their relationship. You can also map them into the common space and link imaging with genetics or map the vision with language, right? And then you can actually, if you have one modality, you can generate the other one, right? So I have an image, I can generate radiology, like a report, right? If you have a report, I can also generate image. And there's also a lot of ethical AI issue, right? Then trustworthy AI, we have to address the fairness and also the privacy, explainability. So I think there are a lot of statistical methods can be used to do these. And finally, the multi-scale, right? You have data from different scale in order to have a better understanding of the biological mechanism. I think the causal inference is important, right? You have multiple things, then mediation analysis, Mendelian randomization. So all these methods actually can be applied to here. Then we can have a better understanding of the neurobiology, right? And then hopefully we can identify target, look for drugs that can modify those targets. Yeah, we have a review paper that talks a little bit about some of these topics if you are interested. And that's it. I want to thank NIH and SF for funding our research, and also thanks to my lab members for their hard work. Thank you.
We leave all the questions at the end.
Okay.
Our second speaker is Professor Wang from Tulane University.
Thank you very much for having me. So I have been enjoying the workshop so much. So it's a great effort by Jen and Hongtu for organizing such a wonderful workshop. So I want to share our experience for integrating multi-scale and multi-modal brain imaging and genomic data in the last few years. So as you know, the use of multi-scale and multi-modal brain imaging and also multi-omics become almost everywhere in neuroscience research. So one of our projects funded by NSF and now by NIH is to study the brain development for adolescents. We propose to collect the adolescent from the age 9 to 15. So we have their multi-modal brain imaging data, including MRI, MEG, and at the same time, we also collect genomic and epigenomic biomarkers to understand what is the underlying genomic mechanism underlying the brain development. So this is a joint effort between 3 other states led by Wiz Hong, Julia Stephen, and Tony Wilson. So our goal is to extract the information from imaging from MEG. We know MEG offer very high temporal resolution. At the same time, we have the multi-modal MRI image, which offers very high spatial resolution. In addition, we have genomic and epigenomic data. So we can extract the functional connectivity network and also some other structural information to integrate with genomics biomarkers such as SNPs. One important feature is about the interactions between genomics and imaging biomarkers, which is considered to be epistasis factors. So there are both interactions within the same modality and the interactions between modalities. We can incorporate such interactions into multi-modal brain imaging and multi-omics, and use the integrated information to make a better prediction. So we develop a lot of models. A particular model we are interested in is co-regularized learning. So the idea is we can use both the regression model and the correlation model and couple them together. So on one hand, we can use imaging and genomic data to fit the phenotypical traits. At the same time, we perform correlation analysis and use this as a prior to incorporate into the regression model. So mathematically, if we have m data sets, n samples, so we can use the regression model to fit the phenotypical trace Y. In addition, we can incorporate their interactions as an regularization term. We can also use prior knowledge such as structural information in the imaging genomic data, which is very typically found in imaging and genomic data. So let's look at two data sets. In this case, if we take U1 and U2 to be X1 and X2, and we can find that the last term actually is a CCE term, and the second term, so we can enforce sparsity or smoothness to consider the structural information in the data. We can further improve the model by dividing the coefficients beta into alpha and theta to account for the regression and interaction separately. In addition, we can incorporate the prior knowledge into the model. So we couple all those information in one framework. So this gives us some advantage for incorporating the interactions between imaging work source and the genomic SNPs. And we can tune the parameter gamma to incorporate such interactions. So this is a linear model, and it cannot incorporate the complex interactions both within the Shen modality and between modalities. To this end, we propose a deep learning version of the collaborative learning. So the idea is we can project the imaging and genomic data using a multi-layer neural network. So this way, we can extract the complicated interactions with the Shen modality. So at the output, Z1 and Z2, we use another CC layer to consider the interactions between the imaging and the genomic modalities. And in the end, we link the data fitting with the phenotypical variable Y, while also considering the interactions between imaging and genomic data. So you can consider this as a representative learning model. So this model actually can include the deep CCA as a special example. Deep CCA only find the complicated interactions between imaging and genomic data, but it does not link with phenotypical traits. So our deep collaborative learning can do both. So we can use the following object function to formulate our deep collaborative learning model. So the first term is actually the data fitting term, the correlation term to consider the interactions between two types of data after projecting using multi-layer neural network. The last two terms are the data fitting term by fitting the deep learning or deep representation of the imaging and genomic data to the phenotypical trace Y. So we can use backpropagation and statistic gradient descent to minimize this object function to train this model. So let me show the application. So we can apply this model to study the brain connectivity network. We know the brain connectivity network can be used as brain fingerprints and can be linked with cognitive behaviors. So here we tested the model on the Philadelphia neural development cohort study. This is a large-scale study containing both multimodal brain imaging and genomic data. So we have 989 subjects, which have 3 imaging modalities, including resting functional MRI, MBAC or memory MRI, and emotion MRI. We do a number of pre-processing steps before running our deep collaborative learning. So here is the result. So we use this model to make a prediction about the 2 groups. One is a pre-teenager group from the age 8 to 11, and the other one is young adults. So using the deep collaborative learning and use the combination of 2 modalities, we can get the best accuracy in predicting the age groups at the 95%. And you can see this model can do better than CCA, kernel CCA, and other regression models. So if we project the high-dimensional data into those representations, you can see using the deep collaborative learning, we can better separate the 2 groups. So they form 2 clusters. So we can easily separate the pre-teenager group from the young adult group. Another question is, can we identify the important features behind the classification or prediction? And we know the deep learning is just like a black box. There is a lack of good interpretability, right? So we just use the occlusion sensitivity analysis. So the idea is very simple. So we can block some functional connectivity network and see without this functional network whether the classification accuracy will drop. If it drops, then this functional network must be very important. For example, for the sensor motor network, if we remove this sensor motor network, so the accuracy drops from 95% to 65%. Therefore, sensor motor network is an important network that contributed to the final classification. So this is one way for identifying the significant brain subnetwork. However, this interpretable model cannot find the feature at the pixel or voxel level. To this end, we recently proposed to use convolutional collaborative learning in combination with gradient cam. So gradient cam is a very popular approach in machine learning that can be used to identify significant features at the pixel level. Take example, here we have the genomic snakes. First, we use some prior knowledge like gene text to do the pre-filtering. Then we run one-dimensional convolutional neural network. Similarly, for the brain connectivity network, we run two-dimensional convolutional neural network. Here we use convolutional neural network architecture because for convolutional neural network or CNN network, we have good interpretability method to help us to identify important features. After we extract the feature map, so we add a collaborative layer to link the genomic feature with the two-dimensional functional connectivity feature map. So at the end, we plug the gradient cam approach to identify significant genomic SNPs and also important brain connectivity network. So here is the loss function used in formulating our convolutional network. Because of time, I skip those slides. So for the convolutional neural network or CE model, so recently there is a very good approach to help us to identify important features. This is called a class activity map or CAM approach. The idea is to find the optimal combination of feature maps based on the convolutional network. And this model can be further improved using gradient CAM. And we also use score CAM to replace the gradient to get rid of the impact of gradient, which is sensitive to the noise. And if you compare the score CAM with gradient CAM plus, and many other interpretable approach, so the score CAM can give a better visualization on the important feature map. So let me skip these slides. So so at the end, we apply our method, to identify important genomic biomarkers underlying different intelligence. And so we found 3 important harbors which show the difference between the low IQ and high IQ groups. So we also identify important genes and perform gene enrichment analysis and link with genomic pathways. So in conclusion, I feel that our statistical modeling can be inspired by the concept and the technology from deep learning field, and the deep learning offers a powerful way to detect the nonlinear connections in the data. So we can combine the advantage and of both traditional statistical model and deep learning model to solve the challenge in neuroscience. So there are a lot of challenges that we are facing. So my group is a big supporter for reproducible research, and to this end, we make the code and the tools to be publicly available, and you're welcome to use the source codes that we published and tested on your own data. Thank you so much. Yeah.
No, your own last speaker, Emil.
Yeah.
Okay. Thank you. And the third speaker is Dr. Bingxin Zhao from the Yale.
Thank you. Work. Okay. Thank you. Thank you very much. It's a great honor to be here to present the research I have been working with. I have been working on, and first I want to thank for the organizing committee, Jen, and especially Jen and Hongtu and the whole group for making this event happen. And today I will talk about one applied project in the imaging genetics. We try to basically in one sentence we try to do the disease prediction or stratification for people without any imaging data. But we want to leverage the imaging data in other data sets. So this is most of the work has been done with my student from Purdue University. It's actually not far from here, like 2 hours drive right from here to West Lafayette, Indiana. So and most of the work has been done with by Shen, and we also have actually the co-author here, Hongtu and Fei and Li. Right? Thank you so much for your support regarding this project. So, and this project also has been recognized by the especially the genetic community. So, Shen actually get 2 awards for this. One is in the GSM this year by the ASA genetics section, and the second one is last year in the ASHG. She also gets the Research Trust Award for this project. So, so this is actually very it's the big motivation of the project is we know the UK Biobank maybe the largest imaging data now, actually literally claim to be the largest one. And why that? Because they collect like 100,000 subjects from doing the project. But this is very big in the imaging community. But if we take a look at from like a scientific community or especially the genetic community, 100,000 is not it's not so big, right? Take a UK Biobank as an example. They have, for example, 500,000 subjects, right? So this is the benefit from the genetics. And if we even see the map they have been working on, I mean, the genetic consultants, they have been working on our effort schedule. They are trying to do this like a global, like a biobank maps, right, from different countries and different research groups. And they are able to collect genetic data at a very lower cost because that's the nature of the genetic data. It's easier to collect and it's much more less expensive compared to image data. So, and the UK biobank imaging data is in this part actually only like 20% of the whole sample in the UK biobank. So, and so then the motivation is we try to say, okay, whether there's some strategies we can really like extend the utility and the impact of the existing like UK biobank imaging study, right? So we get the data from 100,000 subjects, but maybe we can have bigger impact if we can apply this data to more like general cohorts in other research fields. So here is a few facts, like the imaging trait sample size is relatively limited compared to other like data types, and imaging traits they are heritable, right? And we know they are polygenic, which means they can find lots of genetic signals. So the very naive idea is, okay, we try to do this like genetic imputation of the imaging traits, right? So we try, because we can collect data, the genetic data from much larger group, right? So we can try to see whether we can use this genetic marker to impute this imaging trait, right? So that's a big idea. So how to do that? Actually, from a genetic perspective, it's a whole field that I'm pushing forward this. We call it polygenic risk scores because they know lots of complex traits. They are polygenic, they are heritable, and we can do imputation using the genetic marker. So this is one figure taken from a review paper by Shen group from Yize, and they have a very nice review of all these methods already developed for doing the polygenic risk score research. And so then we basically use this method and to design the study. So the first part is the overview. Basically, we have some subjects, especially from the UK biobank imaging study. We have both genetic data and imaging data, right? So basically, we can train the model. Then we have a large number of subjects only have genetic data. Then we can do imputation. Right? And eventually, in this very large cohort with only genetics data, we do get other data types. For example, the disease data. Right. So then this paper actually focused on whether we can do disease risk stratification. So in the middle is like the imaging markers we have been considering. We consider both of the brain imaging, MRI, function MRI, structural MRI, different modalities, and also we have like a whole body image, like heart imaging, abdominal imaging, and even the eye OCT images. And on the right is what I mentioned with the disease stratification. We try to link to disease to show whether we can do the imputation of the genetic scores, right? So then the next question is how we can show they are useful. So we want to find a few angles to tell people, okay, they are useful. So how to do that? Because we check the disease data, the EHR, like health record data. So we want to do a stratification. It's also a typical approach people in genetics they are working on. Basically, they get the distribution of the genetic imputation of the, for example, imaging data or any phenotypes. They want to check, okay, whether we can do stratification in the sense, okay, whether these people have maybe higher risk of getting certain disease than the people in this area. Then if that's the case, they can do in real life, they can do stratification, right? So for the disease risk. So then we basically here is some like results. There's results for the brain MRI data. So we do have some like initiative funding. For example, the Alzheimer's disease, we see lots of like function traits. Actually, they are genetically imputed function traits. Can do the stratification of the Alzheimer's disease, right? Which totally makes sense. But just remember here is we have no real function data for these subjects. We just use their genetically imputed function data can do that. Okay, and we have other like a disease. For them, for diffusion and structure mind, we get lots of signals for the MS. And if we take a look at one signal, it will be like we can see really the trend. X-axis is the quantile that I just mentioned of the distribution, and you can see the risk of on the y-axis the risk of disease. You can see the decay, right? So because we can see okay, this group of people have a higher risk of getting this disease, right? So everything is done by no like image data, it's just like the genetic imputed image data. And we also do it. This is a second example on the heart MRI. So during the time, I can really go to details. So this is another example for the non-brain disease. Is the one disease of the aorta is very serious of disease of aorta, and we are also able to we are also able to check the risk and identify people with higher risk of this disease using imputed heart image data. And we do some like deeper analysis to get a better understanding of these features. For example, the first one is we know these genetic scores will not change in your life, right? Because that's genetically imputed traits, and the genetic risk will not change in your life. Mostly will not change. And then we want to understand at which age it will work, right? And then for the Alzheimer's disease, it turns out maybe at age like 60 or 65, which it totally makes sense, right? Because at that time, it's the age, right? People will get like Alzheimer's disease. But remember, this has genetic predict risk, right? Which means you can get this risk prediction much earlier in your life. Right. So if you do something right before age 60, you might get a chance to reduce the risk. Right. So that's the point here. And we also do some like joint analysis with the disease polygenic risk scores. With basically the key idea is we want to say, okay, for Alzheimer's disease, for given each of these specific disease, actually we are able to directly apply the disease polygenic risk scores, right? And see, we just want to say, okay, we can get additional information on top of the disease risk. So in this figure on the x-axis, different group of people with different level of disease risk. And for this group of people with higher disease risk, basically we can further stratify them with a even higher risk of disease and lower risk of disease. This is done by the imaging data we have. Right, so this is one example, and we have a few other examples in the paper which is already on MetaArchive. If you are interested, feel free to check that. So yeah, this is a summary of the applied project. And so basically we developed this like imaging, genetically imputed imaging traits, right? So it's over 4,000. We already made them available on Benadryl. It's free to download. And so we show they are useful from this angle because we show, okay, we can stratify people with a higher risk. Just use this like you can see like a pseudo image data or fake image data, right? So there's no image at all for these people, and we can use them to stratify disease. So we compare with the disease risk score, we can offer additional insight on top of this disease risk. So now we are trying to work on to test this idea in more like a global, like genetic biobanks. As I mentioned, genetic data is much easier to collect, right? So if we want to show, for example, this is useful in, for example, over us data, right? So which is good news for the whole imaging community, right? Because the imaging data is taken from the UK biobank, 100,000, only 100,000 subjects. It's the only, right? And but they can have much higher impact in other research communities. So then I will have maybe 2 or 3 minutes for 3 questions. So I'll try to answer, but I think it's just some like idea that I want to share. Definitely, it's like this question. Definitely, we try to ask ourselves, right? Maybe every day or every week. Right? Definitely, everyone have their own opinion on this. So the first question, like, what's the most important research problems we want to focus? And so for me, like, I want to like reinforce on if like UK biobank imaging data. So this is the picture. They are getting more sample. I think this participant is the one getting the exact like 60,000 image in the UK biobank study, and now they are launching like as Tom mentioned, right, in the keynote speech, they have the longitudinal imaging data taken from the UK biobank. Still, I think they are very useful resources in the next 5 years. Right? Next 5 years, still they are very, very useful resources. So my one thing will be maybe we can consider how to like making the better use of these amazing data resources and make it more powerful in the scientific community, maybe beyond the imaging community. And this includes both with like 100,000 images will be supposed to be ready in 2 years. The full sample should be ready in 2 years, and the repeat imaging for like 60% of the original samples. And that's of course a lot of opportunity and challenges. So this is my answer for the first one. And for the second one is specifically for the statistical community and how we can improve the process and sharing the data. So when I see this question, I want to ask another question is, so in terms of this like imaging community, who actually does most of the dirty work? Think about we are a team together, like a neuroimaging team. So who does most of the dirty work? Dirty work means, okay, something you cannot get credit, right? For example, this player is doing the screen assistant, right? For this player, but it will not in his statistics, right? But something are not recognized by the society. So I think for me, I feel maybe most of the dirty work actually is done by people not in statistics. Honestly, that's my like intention. Maybe from like people in radiology, in bioinformatics, in bioengineering, right? So they are not done by statistical people. So if now we are thinking, okay, for how the statistical community are sharing the data, maybe one thing is we can play a more fundamental role in this work, right? So in this like neuroimaging team, we can play a more fundamental role in the sense that we are willing to do more dirty work. We are willing to do more like build more infrastructures. And if we consistently doing that, at least of course we will have more like decision, like at least we can make more decision whether we want to process and share the data, which means basically my suggestion maybe is doing a more deeply doing more dirty work. And this may be slightly different from the current like education in statistics, right? So because we always encourage our students to be more to get more smart ideas to do this. But on the other hand, maybe this is also very useful to do some dirty work. And this another side, maybe the community can say, okay, we can get more declaration by doing this. Okay, the last one. So last one is the last one is the question about how the statistical models can be more impactful in the imaging community. So I will this remind me of this like saying from the NVIDIA CEO, like he mentioned that a lot of opportunity in digital biology because they can be engineered, not just scientific discoveries. Right. Then for this question, so my answer will be like it's actually similar to the second one. So maybe we can be more open to engineering work. Right. In addition to be smart, we can also be very hardworking to really can code stuff, and then we can bring our package to the other people in the imaging community. And for us, maybe we can consider how to improve the coding skills, our own coding skills, and also the next generation statistical students, the coding skills, right? So that's a lot of things. And also, I think that's other resources we can use. For example, there's really diverse and rapidly changing need of the researchers in the imaging community. I give one example is maybe last month, I think the UK Biobank just changed their data management policy, which means you can no longer download the data at a certain point, maybe later this year. So they create this opportunity, right? If you want, this every imaging data analysis have to be done in the cloud computing of the UK Biobank. Of course, they want to have very very strong, very solid, like a pipeline of different methods we have already developed online on cloud computing. And also that's power of the social media, right? So you can go to social media to, sell your idea and sell your research plan. Sorry, but I think I have one minute late. Thank you so much, and it's really appreciated. Thank you.
Our last speaker is Dr. Bingxin Zhao from Yale Biostatistics.
So yeah, hello everyone. Thanks Shen, Hongtu, and also organizing committee for organizing this workshop and also for having me. I truly enjoy all the talks and the conversation after the talks. Today I want to briefly share some of our recent work on heritability and the genetic contribution analysis of structural and functional coupling of human brain. Like a background is lots of interest generated recently in terms of study the relationship between brain anatomical structure and functional architectures. Generally, people believe like the structure of the brain constrains and partially shape the functional, like the neural activities and the functional connectivities. Meanwhile, those functional architectures also in turns partially determine the structural brain structures. Particularly lots of consideration is on like the connectivity part, like how to study the coupling effect of this structural and functional connectivity using like a different, under different population, different age range, and people find that it's actually changing across different regions. For instance, this is an example, like using atlas, like a discrete atlas, we can get structural connectivity, functional connectivity for each subject. Then at this brain region I, then we can extract the functional connection, structural connection, and then study their relationship using like Pearson or Spearman correlation. Growing studies suggest that this could be a sensitive and informative neural marker on top of the existing structural connectivity and functional connectivity. However, on the other hand, there's limited study to investigate what's the genetic architecture of these coupling trees. Alternatively, of using the discrete atlas, one can also consider to use the gradients, which are recently growing extremely attention in neuroscience field as a low-dimensional manifold representation. So can study the continuous spatial change. The gradients can be input on different type of trees, but the connectivity gradients definitely is one of the most popular one to describe the continuous transition. So for instance, like shown in this paper, which is gradients of functional connectivity, this is like the first three gradients, which basically just use like a simple dimension reduction, like a PCA or diffusion map embedding. So the first gradients can show like the transition from unimodal networks to more like a transmodal networks. And then the second gradients shows the transition between like more like just an attention network to default mode and so on and so forth. It's like it's very interesting hierarchical spatial architecture there. So on the other hand, I think there's like very limited, or we haven't seen like studies to study actually the coupling effect on the structural and functional connectivity gradients, and without even mentioning what's the genetic architecture for that. So I will quickly discuss two of our studies. The first one is to uncover the genetic architecture of connectivity coupling, and also extensively study the relationship, genetic relationship with different complex trees. And the second one is to investigate this gradient coupling and the genetic contributions. I quickly just describe what we our data is the HCP data, and then we use the atlas, which Zhong already mentioned in his yesterday's talk, which is this continuous atlas, and then because it gave us a finer level of information, eventually we have over 3,000 nodes. And we calculated this structural and functional coupling trees at each node for each subject. And this E is in terms of we can constrain our domain to calculate that. And we use different ones. And then for the genetic data, we have those number of SNPs after QC. So HCP data, given its twin structure and sibling structure, is the ideal data set to study the heritability from those family structure. But if we want to do genetic correlation, then we need to, association, we need to account for this sample relativeness. So this is the heritability part. And the first three shows the heritability for coupling trees, for structural connectivity, and functional connectivity and structural connectivity. And then we also show how it diverse and the different functional system as well as the structural subnetworks. We also show how the coupling trees, the heritability related to structural connectivity and also the functional connectivity. So as we can see, this heritability of the coupling trees slightly more related to the functional connectivity with the correlation like 0.35, but their correlation with structural connectivity are almost zero. So it gives us like some information that cannot be offered by simply study just the structural connectivity, functional connectivity. We have done genetic association, which actually sounds like a really ambitious given the sample size is quite small. And eventually, surprisingly, we identify lots of signals, but given the small sample size, we need to do lots of replication and validation analysis. So we did perform a validation analysis using a slightly larger cohort from the ABCD data and replicate some of our identified signals. And we have also done robustness analysis, like do subsampling of like 90% of sample, 80% of sample, and decrease that to see the agreement of our result. And in terms of the association of the identified genetic underpinnings, as well as our estimation of the heritability. And when the data decreased to 80% and 70%, it looks still fine. And the genetic correlation part, given we do have the GWAS result, we can do this genetic correlation within our trees and with other imaging trees, as well as other complex trees. So as we can see, we did identify lots of significant genetic correlations. So to explain like what this means, like for instance, we detect some positive correlation and negative correlation. So for the negative correlation, for instance, that means like the genetic variance, when it increases this coupling trees value at this brain region, it will induce a decrease of the mode scores for that part. And those p-values, those are significant ones after p-value adjustment. So that's the coupling part, the connectivity coupling part. Then we move on to study this coupling of gradients. So we first, this is just a show like what's the population, structural gradients and the functional gradient, first the structural gradients and first the functional gradients. And then we constructed this structural and functional gradients coupling metric for each of our nodes. And after this is using the diffusion map embedding, and then we use inner product to calculate this coupling. So we then move on to study the heritability of these trees. So here we can see we also just separated this heritability trees under different functional system and the structural, structural subnetworks, and then compare their difference. So we find out that their difference in terms of their different functional system are more significant. We find lots of significant difference after p-value adjustment, but not for the structural one. And this is showing that their difference in terms of their coupling trees architecture driven more from their difference in the functional architecture. And we also stratified by actually also cognition, but I just show sex result here, like male and the female. We find out in this also different functional systems, since they are more distinguished, we find out like in all those different functional systems like default mode and also the ventral attention networks, their heritability for male and female are significantly different. And for all the difference, males have a higher heritability than female. And so we still are exploring other traits in terms of other cognition and other behavior traits to study their this heritability difference. We finally move on to consider again some validation. So given like the MRI, they are non-invasive. So if we want to do this validation, we do some imaging transcriptomic analysis to validate our result. So this is like just a pipeline showing how this procedure actually works, because we can like after we obtain some results and then using say certain Allen brain atlas, and for different brain regions, we can also obtain the tissue for each brain region. And then we can find out if after we parcellate into different cell type, and then we can find out like the gene expression and correlate it with our find out the imaging trace and ranking the gene expression, and find out what genes are mostly expressed based on our study. We did exactly like this and using the Allen brain atlas for lots of genes, like more than 10,000 genes. And this is an example of the top genes. Like this top genes is this ELA-VL4. They are strongly correlated with our traits and with a correlation like 0.41 and p-value. And this genes is like predominantly expressed in neurons and play a critical role in development and differentiation of neural systems. And then we have also done some gene set enrichment analysis and showing that some genes, the genes that are ranked highly there, for instance, are positively correlated with this excitation neural markers, and they are related in they are collected at the deep, typically collected at the deep cortical, like layers are linked with like deep layer neural activities. So those are kind of the architecture of our those those gradient kind of trees we studied. So this is acknowledgement of my PhD student who's in Meta now, and then a bunch of my postdocs, and also my colleagues, and also Zhengwu there, and my grant funding support this. And I have 2.5 minutes, so I can answer those 3 questions. So for the first, 3 minutes, okay? So I can, so the first question is about like the most important questions. So for me, I think like besides imaging genetics, which definitely still has a lot of room, I started to get quite interested in some health-related outcomes. Like so for extract, like say for EHR data, but typically those imaging data, EHR data, they're not linked. So there's a lot of work needs to be done to try to just either study them using some federal learning or just try to put them together so we can study them. And the second question is about improving processing. And I think definitely joining the pre-processing stage, which I actually has been doing in the past few years with a collaboration with our radiology collaborator. And we did find out lots of even though their pipeline seems very mature already, but there's lots of I would say not like that reasonable setup, which we can try to crack. And also just or pay attention in our downstream analysis. And finally, is how to make it more impactful. I think it's more like a willing to educate our collaborator, particularly those scientific collaborators. I fully agree with, I think Ting mentioned in her answering last time. I feel it's impossible to just ask your scientific collaborator to say, okay, what question you have, and then I can answer that using analytical approach. There's no way to do that. This is also my experience is I need to bring up the question. And so after I understand the data, I need to understand the science. So I can say, okay, how about we do this? And then they can tell, oh, whether that's reasonable or not. But I need to be the person first to bring those up. So, and also I feel like just educate them the powerful of the method and then actually showing they can solve some problem and they can bring scientific impact and then they will appreciate. Okay, that's all. Thank you.
Thank you. We'll open the floor for the questions. Yeah.
Sorry if I missed it, but did you control for genetic ancestry in the heritability analyses?
Sorry?
Did you control for genetic ancestry in the heritability analyses?
Genetic ancestry. I don't think I have.
Okay.
Yeah, I think like for both heritability analysis, I purely using an ACE model. It's just those traditional ACE model based on their family structure.
Question for Bingxin. So how much variance in the testing data set that genetic can explain the imaging-derived features?
It's a very question. So it's short answer is definitely it's okay. The short answer is I think you are asking for the prediction accuracy, right? And it's just a small proportion of the heritability we have in the training data, because they are like the polygenic traits. So we can you can think about the heritability as the upper like bound for the prediction accuracy, and now we cannot really fully reconstruct the genetic variance in the training data. And now on average, it will depend on the modality we are doing. The worst one actually is maybe the functional MI data because they are a little bit more noisy compared to other ones. But in the example, you can see even a small proportion of the prediction accuracy, the functional MI data is very useful in terms of identifying Alzheimer's disease. So which means there's no like the linear trend between the prediction accuracy and their utility in real like the disease stratification. It's more related, my understanding is more related to say whether they are this modality is really very sensitive to the disease. For example, we know that the function MI it may be sensitive to Alzheimer's disease, right? You take if we are really able to collect the function MI data from patients, then maybe we can identify each certain like networks. It's not like it's abnormal compared to the highest cohort. Yeah, but in general now still it's still at the pretty early stage of this genetic imputation, and if we are able to get a larger sample size, for example, if the 100,000 subjects are done in 2 years, we might be able to improve. But my guess is we can maybe in 10 years we cannot really get to the heritability level we observed in the training data. Yeah, even 100,000 is not large in genetics if we want to do prediction of the complex traits, especially brain traits.
Yes, supposed to answer questions.
Sorry. I actually have a question for Bingxin also related to Genomics. So you can predict all these imaging traits, but there are multiple those, right? Imaging traits. And are you going to use those together to predict the outcome or just to do that one by one?
Okay, so it's really a very good question. So here for now, we are just doing marginal screening, as you mentioned, just like each imaging modality and each imaging trait separately for each data. I think that's a very, very good idea to put the imaging traits together, even put the disease together. Right? So we can have a cluster of disease, for example, from cardiovascular. Maybe we can see better, more interesting.
Right. I think if you put those together, it's kind of like similar to the concept bottleneck model, right? Because you kind of want to map the genetic variation onto each imaging trait, right? That's more like a genetic component of the trait measurement, right? Yeah. The variance explains, and then if you put those together to predict the outcome, it's more like your model is more explainable because I know hippocampal region how much genetic component expressed, right? The genome expressed at hippocampus and entorhinal cortex, and if jointly, hopefully, I feel in
improve the prediction accuracy, right? If you use multiple of those. Yeah.
Yes, it's a good idea. So we do try a little bit to see whether we are able to, you know, combine a few imaging trees together. We did try that in the early stage of the project. We found that the improvement for now is minimal, maybe because of the training data is not large enough. And we do get one idea related to this. We developed something called like a burden score of imaging trees, which means we can combine multiple trees, but we only care about whether we have some subjects consistently to be at the top 10% or bottom 10%. We try to use that information. Actually, that's something we learned from gene expression literature. Actually, people in gene expression idea, they always try to say whether they have outliers of the whole distribution. Yeah, that things work in the current sample size. But definitely, we always trying to think whether we can put all these imaging modality together, imaging tree together to improve. Yeah.
I also have a question for Bingxin. Thanks for your very nice talk. So you mentioned that the pre-processing is very important. So I'm wondering how sensitive all the tuning parameters in pre-processing step to your results.
It's a very good question. Yeah. So it's a very good question. So now we in terms of like the brain imaging data, we try two different pipelines. One is like the official trees released by the Oxford Big 40 group, and they screening like 3,000 imaging trees, and we do the analysis there, and we also do the analysis from the big KP. We have our like ICE pipeline to process the data, and in terms of the disease prediction, we feel they are pretty similar in the sense that if we can identify certain signal for Alzheimer's disease, we can do it in both of these like RCA-based trees and also these like brain like population-based trees. They, of course, for each disease, we see the different like statistical power in certain sense, but they all like targeting on pretty consistent disease outcomes. Yeah, in general, we feel all these like pipelines, they all are able to capture to a certain degree of the underlying truth of the human brain. Yeah, but of course, different diseases, they might be sensitive to different outcomes. Yeah, thank you.
Any other questions? You have too many questions.
I'm curious. Actually, I have a question, two questions for each. So one is when you calculate the gradient map, is that in the spatial domain or temporal domain? Or the gradient is spatial domain. And another one is for the imaging transcriptomics, right? So Allen human brain atlas only like 6 subjects, right? How do you calculate the correlation? It just correlation, right? You reported the correlation, right? Is the N is kind of a little bit modest, right?
So it's because like for each brain region, and then you can obtain like lots of single cells, and then for the single cell part, then you can get lots of gene expressions, and then there will be tons of gene expression. Yeah.
You have single cell.
Yeah.
Okay. Any other questions? There's no further questions. We thanks all the speaker again. Let's have lunch.